I want to switch over to you. I want to talk through, you know, what are the biomarkers that are most informative to drug development? Um, what's associated with determining maybe success even a little bit earlier? Um, talk us through any roadblocks and how they might be able to be overcome.
Sure. So the great thing is that we in the translational space, like most scientists, stand on the shoulders of giants. And so a lot of the old concepts going back 25 or 50 years, like exposure response efficacy, exposure response safety, PK, PD relationships, really haven't changed. And even when you move into the precision medicine space, let's all be honest, we could go back 20 years ago to the days of Genentech and Herceptin, where they were staining for HER2 in the tumors, and they realized that that's a pretty good biomarker of response to Herceptin treatment. Or, Erbitux and EGFR is the same one. There's plenty of precedence. So moving that into the cellular space, so how many cells do we dose? Do they expand? Do they persist? Can we measure them in the blood? Which, while fancy and genomic based, is really nothing more than a good old fashioned PK assay that in the small molecule world would be run by LCMS. And then in terms of how do we predict outcome, like I alluded to, a lot of companies in the cellular gene therapy space, mine included, look at indications like non-Hodgkin's lymphoma or multiple myeloma, and where the big targets are very well established, like CD19 and BCMA, which then brings us back to how do we stain for CD19 at baseline? How do we stain for BCMA at baseline? Do we see relationships between the baseline expressions of those targets and outcome?
Okay. Um, talk us through a little bit about, when you're evaluating preclinical research and that risk benefit ratio when it comes to um, inherent variability to starting material. What are the difficulties in evaluating that from a cellular therapy perspective?
Well, while I'm happy to talk about manufacturing and release assays, I really think that's probably in Doug's wheelhouse more than mine. I know that once it comes out of our manufacturing group, they've released the product, we put it in. The beauty of working with a cellular product as we do is um, you get a lot more variability in vivo in the subjects than you might with a PK, with a small molecule or an antibody, right? And why is that? That's because the cellular material is a living material that expands in response to antigen presentation and targeting. And so the challenge or the opportunity there is to understand how much antigen is present at baseline. Are your CAR T cells expanding? Are they persisting? And to better understand that dynamic, both in the periphery and down in the tumor. And so, to your point, trying to get longitudinal tumor samples. So, in our case, that would be biopsies or fine needle aspirates from lymph nodes taken at baseline and then longitudinally, or bone marrow taken baseline and longitudinally. You've asked the question, and Doug's highlighted the fact that that's really tough to do,
Mhm.
but a needed component of assay development. And then once you get over that hurdle, now you're in the clinic. It's oftentimes tough to convince the sites to do those assays and send you those samples, which are really important to your modeling and simulation programs to understanding expansion and persistence. But we have to always remember the patients come first. And the reason that the sites are so reluctant to send us serial lymph node biopsies or serial bone marrow aspirates is because if you were a treating oncologist and you had a patient there and you said, "Yeah, I need you to come back again next week for yet another biopsy," somebody who's third line NHL, somebody who's struggling, with their health and their longevity, that's a big ask. And so
Mhm.
I think we as a community bend over backwards to try to find more easily accessible biomarkers, even if they're not as informative, just to try to spare the patients the burden of having another lymph node biopsy or the burden of having another bone marrow aspirate.